Recombinant DNA is the creation of new combinations of DNA segments that are not found together in nature. It is the isolation and manipulation of genes that allows for more precise genetic analysis as well as practical application in medicine, agriculture, and industry. In this figure, this is the gene to be inserted in the plasmid. Here DNA is cut by EcoR1 and also the plasmid is being cut by EcoR1. EcoR1 is the restriction enzyme and it results into sticky ends that are recombined by using DNA ligase. Now this recombinant DNA can be cloned into multiple copies. To make recombinant DNA, first DNA is isolated, cut with restriction enzymes and then ligate into cloning vector such as plasmid. Then this cloning vector is transformed recombinant DNA molecule into host cell. Each transformed cell will divide many times. It will form DNA clone. DNA is extracted from the organism and then DNA is cut into multiple fragments. Similarly, a vector is extracted from bacteria. This is the plasmid, circular plasmid, and then plasmid is cut by the same restriction enzyme. That restriction enzyme is used to cut DNA into fragments. So a similar sticky ends will be formed and they can be ligated using DNA ligase to make recombinant DNA. And this recombinant DNA is introduced into the bacteria to clone and each transformed cell will divide many times. Isolating DNA. First, cells disrupting lipid membranes with detergent, destroying proteins with phenol or proteases. Proteases are the enzymes that degrade or destroy proteins. To degrade RNAs, the RNase enzyme is used, leaving DNA at the end. For plasmid vector DNA, alkaline lysis or boiling cells is used. To get purer DNA from either one or two, crude DNA is fractionated on a thiophosgene gradient, precipitated with ethanol, poured over a resin column that specifically binds DNA. They show the steps, how it is done, and we get a pure genomic DNA at the end. DNA can be cut into large fragments by mechanical shearing or restriction enzymes. These are two methods to cut DNA. Restriction enzymes mostly are used, such as endonucleases, that recognize specific sequence and break the DNA chain from that specific point. Most cut at specific palindrome, such as staggered or sticky ends or blunt end. This target gene is being cut by the endonucleases into sticky ends, and blunt end is the straight end. This sequence shown in the purple color is a specific palindrome that is recognized by the restriction enzyme. The cut donor DNA and vector DNAs must be joined together to form recombinant DNA. DNAs are mixed in a tube. The ends will match up because they are sticky. And DNA ligase is the glue of molecular genetics that will use to create phosphodiester bond between the two DNAs. To amplify the recombinant DNA, it is transformed into a bacterial host chain, that is calcium chloride 2 method or electrical impulse method or DNA microinjection can be used. The cell is divided and the DNA will amplified.